So in this chapter, we will be learning about the beta blockers, beta adrenergic blockers, and how those agents are used in the hypertension and angina, other heart problems. So, and SARs of beta blockers. So there are mainly two types of adrenergic receptors, and beta blockers are part of this receptor, adrenergic receptors, and like one part, the other part of this adrenergic receptor is alpha. So there's two types of adrenergic receptors, alpha and beta adrenoreceptors, and they are all both of them are uh, like follows the G protein coupled receptor signaling pathways. That means they are G protein coupled receptors. Alpha receptors, mainly there are two subtypes of alpha receptors, alpha 1 and alpha 2, whereas the beta receptors has three subtypes, beta 1, beta 2, beta 3. So alpha 1 mainly it is uh, GQ type, which mainly stimulatory depending on the situation, and alpha 2 is inhibitory, GI type. And all the beta receptors are G protein stimulatory, GS type. That means it increases cyclic AMP in the cell. So next, so depending on the location, like in the smooth muscle cells, the alpha receptors have different, like opposite function than the beta receptors. Like alpha receptors contracts the smooth muscles, except in the gut muscles, because in the flight and fight system, the there's less attention is needed in the gut. That means the digestion is not required or immediate in those fight and flight situation, but more energy is required, like in the liver cells more glucose is released and glucose is metabolized, and heart needs to be pumped. So actually, like in the gut there is no need for extra blood supply. So that means alpha receptors mainly increases blood pressure in the smooth muscle cells, while the beta receptors dilates or relax the smooth muscles. That means beta receptors in the smooth muscle mainly lowers the blood pressure. But beta receptors has important function in the heart. So beta 2 mainly has the function of this uh, dilation in the smooth muscle cell, beta 2, and beta 1 is mainly found in the heart. So beta blocker mainly works in the heart with the beta inhibitors, beta 1 or 2 inhibitors. But this beta 2 receptor mainly found other than heart, so in the lung. So it causes the greatest side effect in the lung because of this beta 2 receptor in the lung. So when we take beta blocker non-selective, then it along with the heart it blocks this beta 2 in the lung and causes bronchial constriction. So upon activation, it causes dilation of the bronchial cells, bronchodilation, increases airflow, increases blood pressure. Lowers the increases the blood flow, but upon inhibition, it mainly constricts those lung cells. So as I mentioned, that beta one mainly like more selective drugs are used, and those mainly blocks beta one receptors in the heart and reduces the blood pressure and etc. And agents like the beta blockers mainly slow down the heart rate and reduce the force of contraction, and ultimately that reduces the cardiac output and blood pressure. So depending on the receptor location, like in one part of the body, beta blockers might have a different function, but the overall function is combined effect of the beta blockers is to reduce the blood pressure, because most of the um, beta 1 receptors are resident in the heart, and heart contraction of those um, like inhibition, like stopping those heart pumping and force of contraction mainly lowers the blood pressure. But the other side of the body, like smooth muscle cells, their blood flow is increased. So there's a combined effect. As a result, we get lower blood pressure. So the Natural agonists are adrenaline and noradrenaline. Those two mainly activates those beta, alpha and beta receptors. So as a result, we get different effects. Like when there is a flight and fight response, the adrenaline mainly affects different part of the body differently because of those different adrenoreceptors. Like for example, the different function is see mainly the in a skeletal muscle, like one alpha receptors, like in the skeletal muscle, beta 2 receptors mainly increases the blood flow. That means reduces the blood pressure. But at the elsewhere, the alpha receptors mainly reside, and those alpha receptors mainly show the opposite function because of the activation by the adrenaline. That means in the other places, like in the smooth muscle cells, in those areas, mainly like the alpha receptors increases the blood pressure. So next we'll be seeing the natural agonists, and mostly the beta receptors are like the agonist of this adrenergic receptors. Both alpha and beta agonists are mainly used in the asthma treatment, especially the beta 2 agonists, and the antagonist type of drugs are mainly used in cardiovascular medicine like angina and hypertension, those areas. So natural agonists like endogenous agonists, which compounds of our body mainly activates those adrenoreceptors alpha and beta. We know that adrenaline and noradrenaline, right? So adrenaline and noradrenaline both mainly activates those alpha and beta receptors. Like they have a common ring. This ring system with double hydroxyl group or double phenyl groups with this benzene ring is called catechol. And this catechol ring, and this is an amine, right? This amino group with this like alkyl amine. Sorry. So this is amino group, and there's a two carbon or three carbon there could be. So this is alkyl amine and this is catechol ring. So this is the common structure of the catechol amines like adrenaline, noradrenaline, and dopamine. You will be seeing that. So the important thing is this adrenaline has the same affinity for both of the receptors. Like this adrenaline has same affinity for alpha receptors and beta receptors. But this noradrenaline has more affinity towards alpha receptors than the beta receptors. Then what does that mean? That means that when you look at this structural difference between adrenaline and noradrenaline, you can see there's just one difference. That is this methyl group. Other than this, everything else is similar. R enantiomer, R enantiomer. So hydroxyl group and H group, same position and same category ring. But here in the side chain, N-alkyl substitution, this methyl group is extra CH3. Instead of this, it has like two hydrogen. So this here, one hydrogen is replaced by methyl group. So that means like addition of here at the side chain or N-alkyl substitution mainly affects the selectivity towards the two receptors alpha and beta. So if we can alter here or manipulate or add different groups, then we can like manipulate the selectivity towards alpha and beta receptors. So those mainly gives us the idea like how we can start with the agonist drug, like more selective to the beta receptors or more selective to the alpha receptors. So that was important. The next, so now we will be seeing the SAR of catecholamines, the natural agonists of the beta receptor or alpha receptor. So this is the catecholamine structure, the R enantiomer because of this hydroxyl group, and this catechol ring with two phenyl groups, meta and para. So as this hydroxyl group, this is involved in hydrogen bonding to the receptor. So this one is very crucial. And this R form, this R enantiomer, because of this hydroxyl group, is more active. This R enantiomer is more active than the S enantiomer of the catecholamines. Next, like here at this nitrogen and substitution, like one or two alkyl groups must be there, like R1 or R2, and one R should be there, alkyl group substitution. And if we increase this substitution, alkyl group, like the length, then it will increase the selectivity for the beta receptors. So we have seen in the earlier slide, because of the adrenaline has CS3, and it mainly uh, like the noradrenaline don't have the CH3, and this mainly reduce affinity for the beta receptor. So what will happen if we increase the length, then it will increase the selectivity towards the beta receptor. So likewise here, if we in our agonist drugs or antagonist drugs for beta receptors, then like we can substitute longer R groups here for beta selectivity. So next, so this nitrogen is involved in ionic binding to the receptor. And here in this site, in the side chain, this carbon, if we substitute here with the methyl group, then this will increase selectivity towards alpha receptors, particularly alpha 2 receptors. So next, also note that here, just except this one, the methyl substitution, like except in the other place, like here if you add something, or here if you add more than methyl group or something else, then this will reduce the selectivity for the both of the receptors, alpha and beta receptors. So you can't place here or here randomly. But the exception is like here, if you just add methyl group, then this will increase the activity for alpha receptors. So these both phenol groups are important for the activity, because compounds or natural compounds like the monoamines, suppose monoamines, lacking these two structures such as the tyramine or amphetamine, those mainly have no affinity towards, no or very little affinity towards the adrenoreceptors, but they also affect the adrenergic system by in different ways, other indirect ways, not like stimulating the adrenoreceptors. So, and these phenol groups can be replaced by other groups, which mainly forms hydrogen bonding to the receptor site. So these are the compounds can be replaced in the meta position or at the para position of the benzene ring. So these are the groups: CH2OH, ROH, NHCH3. These are the groups.